interesting. And the last speaker of our session number 2 is Alika Maunakea. He's from the University of Hawaii, and he'll be talking about understanding the epigenomic origins of diabetes disparities in native Hawaiians and Pacific Islanders to enable prevention. Thank you, Alika, and take it away.
Great, thank you so much. Can you hear me okay?
Yeah.
Are the slides up? Okay.
Yep.
Great. I really wish Amber were here actually in person. I have a lot of questions for her, burning questions. It was really wonderful talk though. So aloha mai kako. I'm very honored to be part of this GloMiNe Symposium and share some of our community-based research in Hawaii. In this short presentation, I'll discuss an example of work that we're pursuing to understand and address diabetes disparities in Native Hawaiians and Pacific Islanders. And in doing so, I hope it will offer insights regarding the unique challenges and opportunities of microbial research in Indigenous communities. One major area of interest my lab focuses on and studies is the interaction between gut microbiome and immune cell epigenetics that are shaped in part by environmental factors such as diet and lifestyle to regulate inflammation and metabolic activity that underlie diseases of health disparities such as type 2 diabetes. We apply, like many of you, an integrative multi-omic approach to understand and address these health disparities, in particular among Native Hawaiian and Pacific Islander populations in Hawaii. Now, Native Hawaiians of today are descendants of ancient Polynesians that migrated out of Southeast Asia and navigated the Pacific. No, we didn't arrive haphazardly, randomly drifting on a raft. We intentionally sailed on a double-hulled canoe, what modern outriggers now emulate. Settled Hawaii around 300 AD and developed a sophisticated society that was completely self-sufficient and sustainable, albeit isolated for thousands of years, for over 1,000 years. Now, the first contact from the Western world was made by Captain James Cook of the British Empire in 1778. And in 1810, Kamehameha the Great unified the Hawaiian Islands, and his dynasty later adopted a constitutional monarchy modeled after the UK. Hawaii remained an independent, internationally recognized sovereign nation for nearly 100 years before the kingdom was forcibly overthrown and illegally annexed by the US and at the turn of the century. And it's later becomes the state that you know today. Now, over this rapidly changing period, the Native Hawaiian population suffered a steep crash, largely due to introduced infectious diseases, to the point that we nearly went extinct. We were around 40,000 individuals by 1900. Yet due to waves of immigration from Asia, Europe, America, and interracial marriages, which were very commonplace at the time and continues today, the Native Hawaiian population rebounded. Now, despite this rebound, the population suffered from abrupt and severe ecological changes that resulted in social cultural trauma, as I mentioned, with the introduction of infectious diseases, the overthrow of the monarchy, a loss of land and language and cultural practices, the introduction of modernization, acculturation, and urbanization altogether resulted in forced transition from a traditional subsistence lifestyle to a market-based economy-driven lifestyle, and of course had dramatic impacts to dietary intake and overall lifestyle, health, and wellness of the population. I highlight this here because these drastic changes to the environment are expected to have impacts on the ecological and human microbiomes as well. And Hawaii's population today is actually considered the most ethnically diverse in the nation, with a diversity index of 76% due to the changes in the population during that time. So what I wanted to mention now was that the Native Hawaiian and Pacific Islander population in total make up about 1/4 of the state population. Yet their mortality due to chronic diseases such as diabetes, for example, is over 3 times higher than that of the overall state population. The higher prevalence of diabetes happens at a younger age amongst Native Hawaiians and Pacific Islanders than any other ethnic group by about 10 years on average. Why is this the case? Risk factors such as income and obesity seem to appear to be linked to this, but we're only starting to understand how race and ethnicity may determine social environments and experiences that impact gene function throughout an individual's life. And epigenetic mechanisms, I'd like to argue, might explain these interactions. Now, I want to mention here that although epigenetics is relatively new to the field of biology, the concept that environment impacts health is nothing new. In fact, Native Hawaiians have for centuries not only understood this concept, but applied it to their daily lives. One example comes from our Olelo No'eau, or wise sayings, such as this: "Ipa'a ke kino o ke kiki i ka la'au." It means let the body of the child be solidly built by the herbal remedies. And this was said of Hawaiian healers that would prescribe certain herbs in a diet of pregnant mothers with the idea that the constituents of the herbs would help support healthy development of the child in utero. But the concept actually extended further, and it was thought that by doing so, it would help prevent the child from developing adult onset disorders later in life. So in some ways, I believe we're relearning some of this ancient wisdom using modern science. And I also wonder how this might be connected to the changes in the microbiome. Now back to ethnic disparities. An emerging ethnic difference we are just starting to see emerge in our studies is inflammation, where Native Hawaiians and Pacific Islanders seem to have a higher degree of than other ethnic groups. We also observe that high levels of inflammation seems to be characteristic of diabetes, at least in this population. And in fact, this relates to low socioeconomic status and obesity, consistent with findings from prior studies. Now, what could be the source of these ethnic differences in inflammation? Well, as I mentioned, my lab focuses on understanding gene-environment interactions that underlie health by studying epigenetic mechanisms, which sit at the interface between social and environmental factors and drives our developmental and health trajectories. Now, for those of you unfamiliar, epigenetics is the study of heritable changes in the phenotype that's like clinical conditions such as diabetes that are not due to changes in the underlying DNA sequence. It literally means on top of genetics, and explains, for example, how a single stem cell can give rise to hundreds of different cell types that make up your body while we have the same genetic sequence composition. One major epigenetic mechanism we study is called DNA methylation, which is a chemical modification of specific cytosine bases that make up DNA but doesn't affect the sequence. In normal development, DNA methylation patterns throughout the genome can be highly tissue and cell type specific, which I discovered to be evolutionarily conserved across species. And however, there is also significant variability in these tissue-specific DNA methylation patterns across individuals. Yet it's unclear to what extent DNA methylation variability accounts for race and ethnic disparities. Now, because of the cell type specificity in epigenetic patterns, it's really important to study epigenetics at the cell level to better understand its functional relevance. Now, given the role, the link between inflammation and diabetes, my lab focuses on studying epigenetics in monocytes, which are cells in your body that play a significant role in inflammation. Depending on environmental factors and DNA methylation states, these cells can either become pro-inflammatory, which can promote insulin-resistant conditions such as diabetes, or the cells can become anti-inflammatory, which relates to healthy metabolism. Now, as I mentioned earlier, Native Hawaiians seem to have a higher degree of inflammation than other ethnic groups, which relates to their higher prevalence of diabetes. So we're now pursuing a study to determine whether the variability of DNA methylation in the monocytes might account for diabetes disparities. And we're also measuring how much of that variability might be explained by upstream social and environmental factors using the multi-ethnic cohort study, which is the largest prospective study in the US to include Native Hawaiians and Pacific Islanders that started in 1993 in both Hawaii and California. Now, it covers 3 major ethnic groups in Hawaii, where importantly, blood cells were preserved for over 20 years. Now, we took advantage of this unique resource to examine early indicators of diabetes before the clinical manifestation of the disease. Specifically, we identified individuals that over a 20-year period developed and were clinically diagnosed with diabetes. And in fact, that you can see even when this cohort of 12,000 individuals listed here, Native Hawaiians and Pacific Islanders and even Japanese Americans had a higher prevalence of diagnosed cases for diabetes at 18 and 12% compared to 6% for whites, which was a fairly good representation of what we see at the state population overall. Now, per each of these 3 ethnic groups, we selected 100 individuals that progressed to diabetes, so-called incident cases, and another 100 individuals that remained healthy over that same period, for a total of 600 individuals. We then profiled and evaluated their immune and metabolic activity from biomarkers in the plasma, and carefully selected only the monocyte population from cryopreserved blood cells to examine DNA methylation states genome-wide. All right, so I have to stress that despite the fact that we already, that these individuals over that 20-year period developed diabetes, we already start to see some indicators of that happening before that event. In fact, we see a higher degree of insulin resistance in individuals in the incident cases, as well as an expansion of monocytes in those individuals, now again, before the progression of diabetes. And in fact, if we split the data by ethnic group, we observe that it's in the Native Hawaiian group, really, that's driving the higher insulin resistance and higher number of monocytes that we observed in the incident cases. So we're really starting to see differences in the subclinical indicators of diabetes, again, long before the clinical manifestation. particular Native Hawaiians and Pacific Islanders. And we're still integrating this data to further understand the origins of these subclinical indicators. But our first analysis we completed examined the variability of DNA methylation in these monocytes that I explained earlier. Now, this is unpublished data, and we're still putting together for our paper, but we have identified significant DNA methylation differences between individuals that, again, eventually progress to diabetes compared to those that remain non-diabetic over that 20-year period to our healthy controls. And this heat map is showing these differences where Native Hawaiians and Pacific Islanders are actually drastically separating clearly from the other two ethnic groups, and is a visual representation of what we consider to be an epigenetic signature of diabetes risk. Using principal component analysis of the data, we found that DNA methylation explained actually 47% of the variance between control and incident cases. And interestingly, we found in another study that the monocyte cell population is expanded in clinically diagnosed diabetic individuals. So we believe that this epigenetic signature of diabetes is functionally relevant to diabetes progression. We also identified ethnic-specific DNA methylation patterns across the three main ethnic groups shown here. The patterns are actually, in fact, so specific that it separates ethnic groups fairly well, with Native Hawaiians and Pacific Islanders clustering on one end in the green, whites on the other end in the pink, and Japanese Americans in the middle in purple. Here are a few examples of Native Hawaiian specific patterns of DNA methylation at single sites in the genome. And you can see that in this red group here shows much wider variability of DNA methylation than the other two ethnic groups listed here. And interestingly, using PCA analysis again, showed remarkable separation of methylation patterns between ethnic groups and explains about 15% of the variance between them. But again, you see whites that appear in the blue in one cluster here, Japanese Americans in the gray cluster. The Native Hawaiians are in this orange cluster, as you can see here. Now, while the two other clusters are pretty tightly close together, the Native Hawaiian cluster is actually quite variable, showing that basically this is telling us that the sites in the genome, the methylation patterns are not only ethnic specific, which helps to separate these three ethnic groups in this PCA, but for some reason they are much more variable in Native Hawaiian individuals compared to the other ethnic groups. We also happen to find that these differences were enriched in genes involved in immune function and overlapped with those related to diabetes risk. Now, for the sake of time, I'm not going to go over all the data that we're collecting now, but we think this data is showing that the monocytes in Native Hawaiians and Pacific Islanders that are derived from hematopoietic stem cells are already adopting an epigenetic landscape that is shaping the trajectory of the cell's functional potential towards an inflammatory phenotype that predisposes them towards diabetes. Now, what are the potential origins of these epigenetic differences? To answer this question, we actually had to involve and recruit an additional cohort to study Native Hawaiians and Pacific Islanders to increase not only the sample size, but also to add on other data sets and biospecimens that weren't included in the previous cohort. So in order to do this, we use a community-engaged research approach, which represent true partnerships with community-based organizations that serve Native Hawaiian, Pacific Islander enriched communities, and not only to encourage their participation in our cohort studies, but also to share information that we gather with them to support their health programs, which was similar to what Valerie mentioned earlier. And we help to engage these community organizations, evaluate their programs, as well as collect health information that's relevant for our cohort studies, but also for their communities. And we use this approach along with respondent-driven sampling in order to study, in our study designed to collect social network data around health that includes social network influences on health-relevant behaviors from participants, in addition to other social determinants that might associate with health outcomes. So upstream factors, as well as innate intrinsic factors, and the interpersonal factors in between the two that relate to health. So the impact of social networks on health really isn't new. Indeed, links between social networks of friends appeared to be strongly associated with obesity, which Dr. Chris Hawkins described from the Freemium cohort study in 2007. But several questions remain, including exactly how these influences impact health and whether non-communicable diseases like obesity and diabetes could actually be communicable, which may involve the microbiome, as we actually touched upon earlier today. Now, because of obesity is a risk factor, we are similarly mapping the social networks of diabetes using hemoglobin A1C level. And although preliminary, we are starting to see clusters of A1C among social networks, those with high, low, or normal A1C levels, for example, appear in social network clusters. And interestingly, similar to what Dr. Christakis reported for obesity, we see networks with the strongest similarities between A1C that account for this clustering are typically among networks of friends and peer networks of friends, rather than other relationship types listed. We further found that individuals who interact more, are younger, are friends, and are in particular Native Hawaiian ancestry have a more similar A1C to each other than other relationship types. And with this information, we can start to add on many more questions, such as what's the relationship between the microbiome and these social network interactions, and how do they relate to health? So here's our observed social network, where we again see clusters of similar A1C status on the top. But more importantly, we actually have the gut microbiome data from these networked individuals using 16S base sequencing of stool samples from each of these individuals, so we can compare their profiles. On the bottom network is showing just a randomized network where we rewired these interactions as a comparator. And without going into too much detail about the analysis, we found that there was very similar 16S microbiome profiles among networked individuals, typically of the same gender, who are also strongly influenced each other on eating behaviors in particular, one of the health behaviors that we had looked at. We also observed that prior relationships showing the decreased microbial diversity corresponds generally to health, poor health, including obesity or diabetes. We did observe that in whites in our cohorts, but we didn't observe that relationship amongst Native Hawaiians or the Asian population in our cohort. However, we did find that the variability of the microbiome amongst Native Hawaiians in particular explained more of their diabetic condition than the other ethnic groups. And also, interestingly, we observed that Native Hawaiians showed a higher number of microbial species that were ethnic specific and unique to them. And I'm not going to have time to go through all of the details here. I just wanted to show some of the data. And we also observed that the higher number of significant relationships between these taxa and social variables such as education, income, fiber intake, BMI, and A1C among Native Hawaiians compared to other ethnic groups. And finally, focusing on Native Hawaiians specifically, we found significant differences in the microbiome that related to diet and inflammatory features that corresponded to diabetes in this group. Again, I'm not going to have time to go through all the details, but these data really helped us to piece together a mechanism by which diet and lifestyle factors associated with reduced levels of specific microbial species, including butyrate producers, that appear to lead to dysregulated inflammation via epigenetic processes that are uniquely linked with obesity and diabetes within the Native Hawaiian and Pacific Islander population. And I finally present this work as an example that shows the importance of including indigenous populations in biomedical research, where the higher prevalence of disease conditions coupled with the greater biological variability, ethnic specific differences, as well as the relationships that we've seen with social factors in the Native Hawaiian population really can enable new discoveries, novel discoveries. And this brings me to conclude with some of the potential challenges and opportunities, which was mentioned earlier already. We think that including Native Hawaiian indigenous populations in biomedical research can be a boon to the research, really offer high potential for novel discoveries, as I mentioned. But really, there's cultural, ethical, and social and legal concerns about biospecimen collection, storage, as well as access to information and data collected from the study, as well as addressing some of the more immediate urgent needs of communities that are affected by health disparities, including these indigenous populations. So there are a number of opportunities where we can work in partnership with communities and indigenous populations to really strengthen the relationships by providing a better ways of sharing information and engaging communities in the research process. Valerie mentioned that earlier today. In many of our studies, we promote the care principles for indigenous data governance, which really stands for the collective benefit, authority for control, responsibility, and ethics for data governance from data that's collected from participants. And we also are very focused on increasing the education and career opportunities for individuals in those communities to join the research workforce, because we think we can offer much more than just the samples, but indigenous knowledge is really critical and important for addressing the health concerns of our communities, but also communities that could benefit from this understanding of how the environment is connected to health. And so with that, I just wanted to acknowledge my lab who supported many of these research activities here, and I'm happy to take questions and really glad to participate in the discussion. Mahalo.
Thank you very much, Alika.